Let's talk about sugar addiction and the evidence supporting that it is, in fact, can be addictive. But when you say sugar, what exactly do you mean? Are you talking mostly about glucose, fructose? Does it depend? How do we start to unpack that?
You know, I when I talk about sugar, I'm mostly talking about pretty much anything that's sweet, to be honest. We started off looking at sucrose as the primary sweetener that we were studying, but we've looked at other sweeteners, high fructose corn syrup, and pretty much all the other ones that are out there. And what we're finding from the research is that it's the sweet taste that seems to be driving that addiction-like response.
Mm-hmm.
And so, when we do studies, and we've done lots of studies looking at the brain, what we see happening is that when sugar is consumed, it can release dopamine in the nucleus accumbens and other areas of the limbic system that is the same pattern of release you'd expect to see with something like a drug or alcohol. And you don't typically see that with food. Normally, when an animal eats something, it doesn't release dopamine in the brain. It will if it's a novel food and it's the very first time you've ever tasted that particular type of ingredient or that particular cuisine, because it's more of an orienting response where,
Mm.
it's a survival mechanism. We're kind of built to be wary of new tastes
Yeah.
because when we evolved, it meant if you tasted something funny, it could kill you.
Mm-hmm.
So you better
But there
make
is an
sure you.
aha moment for animals. We're like, I can eat that. It is okay.
Yes. So what's interesting though is that, when we look at the brain, yet most foods don't release dopamine, but when we look at sugar, it does. And it's interesting because it often does depend too on the amount of sugar that's being consumed. When
Yeah.
we're looking at a bolus of sugar being consumed, that's going to release dopamine, whereas, occasional sips of sugar here and there don't have that effect on the dopamine system. So it's almost like we've used the term binging. I've gotten away from that because there's such clinical ties to the term binging. It's just really excessive consumption of sugar that causes this dopamine release. And that's what's happening, for most people most of the time, is that they're releasing dopamine in response to eating those cookies or eating, whatever the substance might be that they're eating. And so I think that's really, a big issue that we need to keep in mind in terms of the fact that, these foods are affecting the brain in this way. And it doesn't stop at dopamine. I mean, we've seen changes in the endogenous opioid system, the endocannabinoid system, again, all of which basically parallel what happens when you look at the brains of animals that are addicted to drugs or alcohol.
Mm-hmm. And so if you put animals in a paradigm, the same paradigm you put them in to assess the addiction liability of a drug,
Mhm.
they exhibit the same basic behaviors in response to sugar. Can you just give us like a concrete example of that?
Yeah. So, are you talking about from like the neuroscience standpoint or from the behavioral standpoint?
Well, let's take them one at a time, but starting with the behavior.
Okay. So yeah, I mean, when we do our studies, like I'll tell you, we started off looking at we had to develop an animal model to study this, first of
Mm-hmm.
all, because when I started doing this work, it was the year 2001. And I had just joined the lab at Princeton, and I was talking with my advisor at the time about what we might study. And, just through some discussions, this kind of came to be to ask the question, well, could food be addictive, just like drugs and alcohol are addictive? And it came out of the fact that obesity, we were starting to hear more and more about it in the media as a public health concern. And, to me, it just didn't seem that it could all be about personal responsibility and it could all be about, willpower, which was kind of what the mantra was back then. And looking at our changing food environment, we started to kind of see, well, there's a lot more sugar in our environment than there was 30 years ago. And now there's a lot more obesity than there was 30 years ago. So perhaps that has something to do with it. So when we developed this animal model, we basically wanted to assess, whether or not binging, withdrawal, and craving, which are kind of like the triad of behaviors associated with addiction, would emerge in response to animals that are just binging or given daily access to large amounts of sugar.
Mm-hmm.
And so we gave them a sugar solution. They always had water to drink too, so they were never forced to drink it for hydration. They always had food available, so they were never forced to consume it just for nutrients. And what we ended up seeing was that, in fact, the animals that had this extended access to the sugar solution would develop binging. They developed signs of tolerance. So, they would have larger binges throughout the day. They'd also show signs of withdrawal. And this was measured using measures of depression. We also used the elevated plus maze to assess anxiety. And we also had somatic indications that we were assessing, like, changes in blood pressure, changes in body temperature, and even just like ultrasonic vocalizations of distress, essentially,
Hmm.
that they can make to each other that we can pick up using different devices. And then lastly, the craving part that really, can be measured in animals. Even though they can't tell us they're craving something, we can do different experiments to see how hard they're willing to work to
Hmm.
get access to a substance. And that's usually an indicator that they're craving. So we used operant conditioning to do that. And, essentially what happens is that, we're able to demonstrate these behaviors in animals that are having this excessive consumption of sugar, but not in animals that are eating regular rat chow or animals that are having sugar just occasionally.
Hmm. Yeah, and I guess one of the keys here is that, you said they always had access to plain water, they always had access to other food, so they can opt in to doing this. And even though they
Yes.
have that option, and it's not necessary for them to hit their nutrient or water requirements, they often will choose to do this, and the binging becomes more frequent. It becomes bigger, and you can essentially measure things like a mouse sugar hangover.
Yeah, basically that's what we've been doing. And, also in people too, because there's been some scales that were developed and ways in which a lot of the work that we started off doing in our animal models could then be translated into humans. And so many of these things can now, and again, with the use of neuroimaging techniques, we can basically see this happening in humans as well. And I think one of the things that, for me is so exciting is that we're able to, capture a lot of what's happening in the brain, because a lot of times behaviors, yes, they are indicative of what's going on in the brain, but to be able to really see these neurochemical changes happening in real time, because a lot of the techniques that we were using at the time of these initial studies were in vivo microdialysis techniques. So that basically allows us to cannulate the rat in the brain and extract extracellular fluid while the animal is awake and moving. So they're basically running around the cage tethered so that we can collect the sample from their brain, and they're eating, they're drinking, they're, engaging in whatever behavior they like. And so to be able to see the rat drinking sugar and then look over on the screen and see a spike in dopamine, I mean, it's really telling to see that
Hmm.
happen.
Hmm. Yeah, so basically, you see all these behavioral signs that the animal is in fact becoming dependent or engaged in compulsive behavior, and then you're saying basically what you've said so far is that you can see in the brain that the consumption of sugar by rodents is eliciting the same types of effects and the same parts of the brain that you would see in response to an addictive drug.
Yep, absolutely. And, when you say it like that, it kind of sounds like, oh yeah, that makes a lot of sense. But I have to tell you, when we first were doing these initial studies, like when I was a, early grad student and I would go to conferences and present some of our new stuff, it wasn't well received. And it was really just, it's really been over the past, I'd say 10 years or so, that this concept has really gained a lot of traction. And I think, again, it has to do with the fact that, we used to silo things so much. Like addiction was over here, obesity was over here, and nobody really put the two together. But I think now we've gotten a bit more integrated in our thinking when it comes to medicine. And, now like addiction medicine is something that people can become certified in.
Mm-hmm.
And apply that to obesity and apply that to treating individuals who are struggling with their body weight. So I do think that we need to keep in mind that, yeah, I mean, there's been a lot of changes that have happened that have, I think, allowed us to gain more acceptance, and hopefully that'll continue.
Mm-hmm. And so, in the experiments you've described for animals, are we talking about when you say sugar water? Are you talking about sucrose here?
Yeah, so our studies initially, and again, because we were trying to be careful scientists, we wanted to make sure that when we were using this model to test all these different aspects of substance use disorder or addiction, that we were keeping things aligned. So we were using typically a 10% sucrose solution.
Mm-hmm.
And that's about the concentration that you would get of sugar in a can of soda if you were to
Hmm.
have.
Hmm.
So that was where we started. And like I said, we've kind of looked at other sweeteners, and we've published looking at high fructose corn syrup. We've looked at glucose. At the end of the day, it really does seem to boil down to that sweet taste. One of the studies that we did, we used sham feeding combined with in vivo microdialysis. So what we did was we cannulated the stomach of the rat so that when they would drink the sugar solution, they wouldn't digest it. It would simply exit through the stomach, through a tube. And when we were to do microdialysis on those animals while they were sham feeding sugar, so they could drink it, taste it, but not digest it, we saw that it was causing a release of dopamine, even though they're not digesting it. So just simply tasting the sugar was
Yeah.
enough to
Yeah.
elicit that dopaminergic response. And even with other sweeteners that have been tested, the whole sweetener world is a little tricky when it comes to rodents because some rodents don't even detect the taste of certain artificial sweeteners just because they're so artificial. They like
They
they
don't
don't
have like receptors for it.
recognize. Yeah, they know to stay away. But yeah, so to me, it's really more about the sweet taste. Now,
Hmm.
it goes beyond the addiction, right? I mean, obviously, there's certain types of sweeteners, like fructose is going to have more of a negative impact on the liver than maybe other types of sweeteners would. So there are differences for sure in
Right.
terms of how these sweeteners affect the body. But just coming from
Yeah.
the addiction piece, it's really
Yeah.
about the sweet taste.
But yeah, so what you're saying is if you sort of regress all the variables here, it's the level of sweetness is proportional to the level of seeking that you're going to get from the animal.
Absolutely. And I think that's when we look at this from the public health perspective, I mean, that's the problem is the level of sweetness has just been slowly getting ticked up. And if you look at the amount of sugar that's in all the different food products that are on the market, it's so much more now than it was 5 years ago, 10 years ago, going
Hmm.
back, you can really see it. And then just the sheer number of products that have added sugar has gone up. So there's just, I like to think about this from, what are we going to see in 20 years? Because, I mean, there's sugar in so many of the different things that kids are eating.
Hmm.
They're basically being raised to have this high level preference for sugar that, basically means that they're going to desire to have things be really, really sweet. So it means they're going to be consuming more and more of it over time.
Yeah. And, one of the things I want to ask about here too is, well, if I step back for a second, I think about sort of why all of the biology is set up this way and, how things work under naturalistic or ecological scenarios. If I think about addictive drugs, it's pretty easy to think about. It's like, okay, the brain needs to have mechanisms to motivate the animal to do things. It needs to have natural forms of reward processing to tell it like, go for this, don't go for that, develop preferences, develop motivations. And it's not surprising that, you see substances like synthetic compounds that have supersized effects in this natural reward circuitry in the brain, whether that's a psychostimulant or an opioid or something. You're like, oh, okay, it's tapping into natural mechanisms that are for reward learning, but it's got a supersized effect effectively because it's not ever anything that an animal is going to encounter in nature.
Right.
And when it comes to something like sugar is of course something that animals encounter in nature. And so, but they're still developing these addictions, as you've just told us. Is the way to think about this that in nature, the dose and the frequency with which an animal will encounter something like a natural sugar reward is limited, whereas in artificial settings concocted by humans, that's not the case?
Yeah, absolutely. And, if you think about it, if an animal or even us as humans, if we were to wander into an apple tree, right? Are you going to eat the whole tree worth of apples? No, we have satiety mechanisms built in where, there's fiber in the apples and we get full from the calories. And so our jaws get tired from having to masticate the apple to chew it up.
Mm-hmm, mm-hmm.
So we have these sort of built-in brakes, if you will, to slow us down, to stop us from over consuming. So that limits the amount of sugar that we have. And also just nature in and of itself. I mean, the world isn't covered in apple trees. You have to find one. It has to be during the proper season. It has to be, something where the apple hasn't fallen off the tree and it hasn't spoiled. It has to still be, safe and attached to the tree
Hmm.
and something that you
You
should
have to do
eat.
work to find it. You might have to fight for it. You've got to chew
Yeah,
it. It's not a liquid bolus of straight sugar.
exactly. And I think that's, really what we're facing right now is that, a lot of the work is taken out. I mean, again, it's wonderful that we have refrigeration and we don't have to, plant our own apple trees, but it does mean that, we get access. And the amounts of it, I think, in terms of taking an apple, let's say, and then, making apple juice. Yeah, you can, how many apples would you need to make a glass of apple juice, right? I mean, that's quite a few apples. No one's going to sit there and eat all those
Hmm.
apples.